Okay, so 8.4, brain development and the environment. Early life experiences shape brain development. There's a loss of plasticity in the brain in adulthood, but there is still some plasticity. But of course, early brains are very plastic and are constantly changing based on these life experiences. When one brain area is damaged, the function of that area might be taken over by other brain areas, and this is especially likely to happen when a young brain is damaged. So, for example, when the Broca's area, which again is this area in the frontal lobe that's associated with language production, if Broca's area is damaged, a patient may regain some of the language function when other adjacent cortical areas start to take over the function of Broca's area. So they might go through a period of not being able to produce language fluently. However, over time, they may regain some of this function due to adjacent cortical areas taking over that function. More experiences and a more stimulus-rich environment are associated with higher intelligence in general. Experimentally, we see that rats with a richer early life environment are better at maneuvering through a maze. So I think the book gave you an example of a scientist keeping some rats in the lab, which was a pretty unstimulating environment, and then keeping some rats in his kitchen, which was stimulating because his wife was chasing them with a broom, etc. And they were just getting more exposure to different experiences and stimuli. Observational, there's a very famous and tragic study of children raised in Romanian orphanages in the 1970s. So there was a rather tyrannical communist rule in Romania in the 1970s, and I think birth control and abortion were illegal, and there were a lot of children in orphanages and not a lot of money to take care of them. So these children in these Romanian orphanages were raised, many of them in one crib. They weren't being picked up, they weren't being held. So it was extreme negligence, essentially. And scientists found that these brains of human children in extreme neglect are smaller. And the children have a lower IQ overall later, and in general just don't fare as well as children who have a more rich early life environment. So you can see over here to the right, this is, I don't think it's necessarily from the Romanian children's study, but this is an example of a brain of a normal 3-year-old child, and then the brain of a 3-year-old child who was raised in extreme neglect, and you can see how small, how much smaller it is compared to the 3-year-old child, and you can imagine how that will follow that child throughout their life. In addition, we see different neuronal architecture in rats who have more stimulating environments during their development, more dendritic branching, more connections between, more synapses and connections between neurons. And we also know that specifically tactile stimulation is very important. So there are a number of studies where baby rats that were raised in a lab were like brushed. They literally used like a paintbrush to sort of pet the baby rats to give them sort of a standardized form of affection, I guess, or tactile stimulation. And both motor and cognitive functioning was improved in the rats who were brushed as compared to the rats who were not brushed during development. And this speaks to the importance of holding babies. So going back to the children in the Romanian orphanages, those kids were not being held. There weren't enough resources, people didn't have time, or perhaps didn't understand the importance of just holding the babies once in a while, and that could have had a really large impact on their later functioning. So for our music lovers, it does appear that early exposure to music affects neurons in the auditory system. There's this particular study where there were structural changes after just 15 months of musical training in children, where there was, there's increased gray matter. If you look down at the pictures on the right, on the left, increased gray matter in the primary motor area for people who played instruments versus controls, increased density of the corpus callosum, so that would be white matter. If you remember, that's the connections between the two lobes, so increase white matter in the tracts that connect the two lobes, and then increases in the primary auditory area. as well. And this was anatomical MRI as a method, which shows differences in the amount of gray matter, in this case, differences in the white matter as well. Early exposure to music appears to affect neurons in the auditory system as well, so connections between neurons. This particular study, Steele et al. 2013, shows diffusion tensor imaging, which is a method that studies the connectivity between brain areas, or the white matter tracts between areas, rather than just looking at the volume of gray and white matter. It's looking at the strength of connections, how many white matter tracts are connecting these brain areas. This study suggested that there might be a sensitive period from ages 6 to 8 in which music can be particularly influential on the development of motor skills. And so they found that kids they looked at the brains of musicians, and individuals who were learning during the ages of 6 to 8 had particularly strong connections in these areas of their brain. So looking back, I think we're looking at the same areas actually as the previous study, so they're not labeled, but this is the motor area here. You can see there are strong connections, very strong connections. The red and the yellow indicate strong connections. In the motor area of these individuals who learned music ages 6 to 8, again in the corpus callosum in the second picture, and then again in the temporal lobe, the auditory cortex as well. Sensitive period is a little bit different than critical period. The idea of a sensitive period is that the brain is most primed to acquire a skill during this time. It doesn't mean you'll never acquire the skill if you learn outside of that time, so it's not as strong as a critical period, but it's still a time during which the brain is particularly receptive to learning a specific new skill. So there are prenatal events that can affect brain development. In fact, most prenatal events can affect brain development. Fetuses can actually hear sounds at 30 weeks and can recognize a mother's voice in utero soon after they start to recognize sound. So there's been a number of studies about this, but there's this one particular study where researchers had a baby listen to their mother's voice reading a poem compared to another woman's voice reading a poem at the same volume. And the baby's heart rate quickened when they heard the mother's voice reading a poem. So they actually had a recording of the mother's voice. The mother wasn't reading it out loud. And this, along with a number of other studies, make a pretty compelling argument that the baby can recognize its mother's voice, can make a differentiation between another person and the mother's voice, even while still in the womb. Unfortunately, prenatal events can have a very negative effect on development in a baby. So teratogens are any agent that causes abnormal development due to prenatal exposure. This includes maternal stress. If a mother is under a huge amount of stress, large amount of stress, prenatally, this can affect the baby's development. Drugs, alcohol, tobacco, cocaine, for example, can have a big effect on development. Can have pretty profound and devastating effects on development of a baby. Lack of oxygen for a period of time can negatively affect development. And the degree to which teratogens affect the development of a baby depends on when they're exposed. So different functions, different parts of the brain and the body are being developed at different times prenatally, and based on when the most development is happening for a given part of the body, so with the hands, for example, or the face, teratogens can have a more devastating effect during periods of faster growth, for example. So fetuses that are exposed to teratogens during development of the midline of the face, which is like the nose and the eyes, it's rather rare, but they can actually get cyclopia, which is comes from cyclops. So they can be born with one eye in the middle of their head. This is actually a really important time for brain development too, so typically these babies unfortunately do not survive. This would result in a miscarriage. But sometimes animal babies survive that have cyclopia. And I spared you the pictures on the slide, but if you want to look online, you can type in like cyclopia kitten, or I think maybe like baby goats are often tend to be born with cyclopia. So you can see the kind of the devastating effects. Interestingly, the same teratogens that can cause cyclopia, because they affect the development of the midline of the face, they can also cause two-headedness. So exposure to teratogens during the same time can cause kind of the opposite effect, which is to create to split the head in general and create two different heads. And so again, I've spared you pictures of this on the slide, but you can, this has happened in kittens and other animals. And it has happened in people too. Although I'm not sure having two heads, I'm not sure if people would survive. There are like twins that are connected such that it looks like they have two heads, but I don't know if one person who has two heads would necessarily survive. More minor issues that can happen due to exposure to teratogens are minor physical anomalies, and these are small physical differences, mostly in the hands and the face. Also happens in the feet, that indicate abnormal development during specific months. So if you look to the right, there are minor physical anomalies that may happen from exposure to toxins during the first trimester. And this includes like kind of a tongue that has like big lumps or is really rough. You see the hand there, there's like they call it like a single transverse palmar crease, so really, really strong single crease in the hand. Ears that look malformed. This in the eyes, they may have eyes for a Caucasian baby, like eyes that are not that cover up the tear duct, where you can't see the tear duct, and then feet where the big toe is separated from the second toe quite a bit, so like an increase in the toe gap between the first and the second toe. Notably, this, like if somebody has these physical features, it doesn't always come from teratogen exposure in the first trimester. These are just, it's kind of a syndrome, a series of physical anomalies that you might see if somebody has exposure to poisonous or developmental abnormalities in this first trimester. These minor physical anomalies are associated with a number of psychological disorders, including autism, schizophrenia, and Tourette syndrome. And the idea is that because we see these increased physical abnormalities that are associated with teratogen exposure in the first trimester, because we see more of those in people with these psychological disorders, there's probably something that's happening in the brain too when people are exposed to teratogens in this first trimester that's making them more likely to have these disorders. So it's highlights the possibility that this might have happened to somebody with these abnormalities. Okay, hormones and brain development. Of course, the sex of the baby is mostly determined by genetics. There's the XX chromosome that would normally create the female sex. XY typically creates male. As many of you know, there can be situations of XY females and a number of different chromosomal abnormalities can happen that result in male or female sex or something in between. Interestingly, the female brain is the default. The male brain is sort of the female brain that's exposed to testosterone. So the Y chromosome steps in during development and creates gonadal tissue, or develops the gonadal tissue into testes. Testes in turn secrete testosterone, which leads to the development of male reproductive organs. And then testosterone also affects the brain and creates some differences between male-female brain, as such as where the neurons grow, when they die, synaptic activity. A lot of basic processes can be different in the male versus female brain. If you look up on the image here on the right, the orange areas are areas that are generally larger or thicker cortex in females versus the green areas, which are generally larger in males. And again, these are broad generalizations. It doesn't mean it's the same for every male and every female. Okay, gut bacteria and brain development. So we've talked previously about the enteric nervous system, or the ENS. And part of this, a very important part of this, is the microbiome, which is gut bacteria. The microbiome is inherited from the mother, typically during vaginal birth and skin contact. So in the case of C-sections, where the baby doesn't pass through the vaginal canal, there's some people believe, and some research backs this up, that babies are not inheriting as much of the microbiome if they're delivered during C-section, and this could lead to problems later on, both problems within the enteric nervous system and within the nervous system in general. There's a risk of some disorders that's associated with abnormal development of the microbiome. So, for example, autism has the microbiome in autism has received a lot of attention, and there seems to be some indication that abnormalities in gut bacteria may, in some cases, be a risk factor for developing autism. And there have been experimental demonstrations as well of associations between behavior and microbiome, where people with behavioral disturbances are given kind of an infusion of a to improve their microbiome, and behavioral problems have improved. And so this is a lot of this research is still in the very early stages, but it's very interesting and from my perspective, very promising in terms of developments that may happen in the near future. Injury in brain development, there's sort of a time during which injury can be most devastating. So the worst time is in the last half of pregnancy and the first months afterwards. So if you've ever heard of like someone dropping a baby on their head, especially a newborn infant, this really can be very devastating. I mean, it can be bad for any kid, but especially devastating for a new infant. In later parts of development, plasticity of the young brain can make it more resistant to injury. Drugs, many prescription drugs, even common prescription drugs, can be harmful to a fetus, harmful to development. The FDA in the US has classes of drugs for pregnancy, indicating whether women should, giving women some information about whether or not they should be taking these drugs during pregnancy. And so a case study of the effect that medications can have on brain development, there's the very tragic case of thalidomide. This was a drug that was marketed in the 1950s for a number of things, but in Canada and Europe it was marketed as a cure for morning sickness. In the US there were some cases, but I think it was never fully approved by the FDA, so it wasn't as devastating in the US. At the time, many scientists believed that drugs couldn't cross the placenta, so not as much attention was paid to medications that women were using during pregnancy. Unfortunately, it turns out that thalidomide is a teratogen, and it caused at least 2,000 infant deaths, 10,000 birth defects, I think 5,000 of which eventually died soon after. And the birth defects were rather severe, so they caused serious malformation of the limbs, sometimes just a lack of formation of the limbs at all, deafness, blindness, cognitive problems, just a whole range of issues. You can see on the bottom right, here's a little girl who was, they called them thalidomide babies that have been affected by this. And she didn't develop arms at all, has adapted quite well to playing with toys with her feet. So interestingly, damage was very dependent on the time of exposure. So if thalidomide was taken by the mother around 20 days after conception, there was central nervous system damage. If it was taken 21 days after, eye damage, 22, ears and face damage. And you can imagine that people were taking it multiple days, so multiple things could be affected. And of course, there was variation across individuals. If it was taken after 42 days, there was minimal to no damage. So it was if it was taken after this certain period in development, it didn't affect the babies as much. And as tragic as thalidomide was, it did bring attention to the effects of drugs on fetal development, and people have been much more careful to make sure that medications have been tested before they're okay to be used on pregnant women. So other sources of abnormal brain development, there are internal sources, things that just happen kind of naturally. One of these cases is spinal bifida. Spinal bifida is when the spinal tubes do not close completely, and a lot of times there's a abnormal formation of the spinal column in general. It can look, it's pretty sad looking. There's usually just kind of like a hole in the back of the spine. Again, you can look it up, but I spared you the picture. It primarily comes from non-genetic causes, and this is one reason that pregnant women are advised to take folic acid, which is a B vitamin. As well, many grain products, such as cereal and bread in the US, are fortified with folic acid in large part for this reason. Folic acid plays a role in cell division, so normalizing cell division throughout development can help prevent spinal bifida, and scientists estimate that a large amount of spinal bifida cases can be avoided just through supplements of folic acid during pregnancy. Effects of abnormal brain development can be subtle, with the full effects emerging later in life. So schizophrenia is a really good example of this. Schizophrenia is considered by most to be a neurodevelopmental disease, meaning that it's a disease that is caused by abnormal development of the brain. And this abnormal development typically begins, at least in part, in utero, although the symptoms don't emerge until much later. So schizophrenia is considered to be about 50% environmental factors, 50% genetic on average. And most genes that are linked to schizophrenia code for brain development, particularly synaptic plasticity, transmission, and functioning of the frontal lobes and hippocampal regions. Typically, children don't show huge changes that would indicate that they're going to develop schizophrenia later. It's usually more subtle behavioral and motor and emotional issues. So kids might have depression, they may suffer from attentional problems, or a lot of times just kind of be like a little bit unusual, maybe socially awkward, and just a little bit different. And then in their mid to late teens, they may start to develop some of the psychosis symptoms, and typically it comes out completely in late teens, early adulthood, with it happening just a little bit later in women, where like early 20s to mid 20s is more typical for women. And this is likely in part due to the later stages of synaptic pruning and the frontal lobe development happening later in life. So as we talked about before, frontal lobe doesn't continues development into the 20s, and schizophrenia, frontal lobe is implicated in the pathogenesis of schizophrenia, and so the later development of the frontal lobe is probably one of the main reasons that schizophrenia emerges so much later in life, even though many of the things that happen that create a risk for schizophrenia happen early in life. Developmental disabilities, obviously there's a range of developmental disabilities, but in general, for developmental disabilities, you see less dendritic growth and fewer spines. So there can be a range of causes. This can be birth trauma, so oxygen deprivation during birth, which can lead to cerebral palsy. It can be malnutrition, mother was not getting adequate nutrition during pregnancy, or baby is not getting it during early years. Drugs, as we talked about before, environmental abnormalities, so like the sensory deprivation, like the tragic case of the kids in Romania, genetic abnormalities, of course, such as Down syndrome, where they have the extra chromosome, and then prenatal disease if the mother gets an infection, such as rubella, which is like the German measles. Rubella has been linked to, a woman getting rubella during a pregnancy has been linked to birth defects later on, and developmental disability.